Howdy, Bio 201 IHers. Welcome to Module 2. We're still in Chapter 3. We're talking about cells. This is the third video in Chapter 3 cells video. And this video we're going to talk about cell transport. Remember, I think there's either 5 or 6 videos for cells because cells are kind of a big deal. They're kind of important. So we're going to talk about cell transport. All right, so when you talk about cell transport, you can talk about a lot of things. You can talk about transport of things around the inside of the cell or in the cytosol. Or you could talk about transport of things in and out of the nucleus through those channels in the nuclear membrane. Or you can talk about transport of things across the plasma membrane of the cell. So a little review on the plasma membrane, or as this is titled, membrane transport. So here's a cartoon of our cell. Remember, this whole thing is the plasma membrane. And this is really important to note. So you could ask, what is this thing called on the outside here? You could say, it's the plasma membrane or the cell membrane. And then a second question you could ask about that is how or what is the plasma membrane made of? How is it constructed? And then you would answer that question differently. You would say the plasma membrane is constructed as a phospholipid bilayer with the polar heads of the phospholipid molecules in contact with the fluid portions. So it would be in contact, the polar heads of the phospholipid molecules would be in contact with the ECF, and the second layer of the bilayer, those polar heads would be in contact with the ICF. And we talked about why that's the case. So the polar heads are hydrophilic, so they love water. The tails, the non-polar tails in the phospholipid bilayer here, they are hydrophobic, so they're afraid of water, so they turn away from water. When these bilayers form, the polar heads move such that they are in contact with water because they're hydrophilic, they love water, and then these hydrophobic tails get as far away as the water as they can, so they form this middle portion of the phospholipid bilayer. Okay, so now we'll talk about transport of things across the phospholipid bilayer. So if you are a molecule, let's say out here in the extracellular fluid, or you could say the interstitial fluid, you could say that. Because remember, the extracellular fluid is made of 2 compartments. One of the compartments, if you are extracellular fluid, you could be blood. And the other thing you could be, if you are extracellular fluid, is interstitial fluid, or ISF. So sometimes the cartoons will just call this outside of the cell membrane, they'll just call the whole thing the ECF. But really, I would say more specifically, because there are no blood vessels out here in the ECF, if I were drawing this cartoon, I would call this the interstitial fluid. Okay, so let's say you're a molecule here, and you want to get from the interstitial fluid through the phospholipid bilayer and into the intracellular fluid of the cytosol, or the cytoplasm. How do you do it? Well, if you are a really tiny molecule, without any polarity, you could pretty easily just diffuse across the phospholipid bilayer. But if you are a big molecule, let's say with a whole bunch of negative charges on you, you couldn't get through the phospholipid bilayer without help. But you could, if Mother Nature had supplied you with a channel like this guy right here, then even though you're a large negative molecule that would never make it past this hydrophobic inside of the phospholipid bilayer, you can get through this big purple channel. So that's kind of an overview of membrane transport. There's two basic methods of transport that we'll be talking about. The first basic method of transport across the cell membrane is a passive process. That means a passive process means that you are a molecule and you will diffuse down your concentration gradient. And that means, let's say if you're this molecule over here, this negatively charged molecule, if there's 10 of you out here and only 5 of you on the inside of the cell, then you have a greater concentration outside at 10 than you do inside at 5. And if this channel is open, you will diffuse down your concentration gradient passively. Passively means there's no ATP needed to get you, negative molecule, from the outside of the cell to the inside of the cell. The other basic method of transport is called an active process. In that case, you have to provide energy to move across the cell membrane. Now, we can use the same example, only this, if this is an active process, what I would do is I would say there are 10 big negative molecules on the outside, and there's only 5 big negative molecules. There's 10 negative molecules on the inside, 5 negative molecules on the outside, and you want to go from 5 to 10. Now that you're not moving down your concentration gradient because 5 is smaller than 10. So the concentration gradient would have you move down it, and that would mean moving from 10 to 5. But if the cell needs to move in this direction, 5 to 10, you're going against your concentration gradient. That means you need your old friend ATP. It's a really important biochemical concept that you need to focus and meditate on. Get your Zen candle out. Go, think about what I just said. All right, so let's talk about active transport. Here's our old friend, the plasma membrane, aka the cell membrane. What's it made of? A phospholipid bilayer. What are the phospholipids made of? Polar heads that love water and are in contact with water, and then nonpolar tails that are hydrophobic. They're afraid of water, so they're in the middle. And now here's this pump is really important to understand. It's called the sodium-potassium pump. Sometimes it's called the sodium-potassium ATPase. Sometimes it's called the sodium-potassium ATPase pump. But anytime you see the word pump in physiology, you should think that you need some ATP. So in this case, let's check it out. How does the cartoon show us? It looks like in this part of the cartoon, it's got 2 sodiums here in the pump, and another sodium is coming from the intracellular fluid to move into this third space on the pump. Now, why do you need ATP to move sodium from the intracellular fluid to the interstitial fluid? Well, if you look at the cartoon, there's some clues. There are 3 sodiums out here, and in this part of the cartoon, let's just say there's 1 sodium here. So sodium is being moved, is being pumped this way, so from the intracellular fluid to the interstitial fluid. That means sodium's going against its concentration gradient, because remember, you have a bunch of sodiums on the outside and only 1 sodium on the inside. That means the concentration gradient, if there's a channel open, would tell sodium to move down the concentration gradient, but your cell wants to move sodium against the concentration gradient, so it needs ATP. That's active transport. And so in this cartoon, it shows ATP gets hydrolyzed. That means one phosphate is taken off, put here, and then a whole bunch of energy is transferred, and you're left with ADP. But what you do is you're pumping sodium against its concentration gradient from the intracellular fluid to the interstitial fluid. And then at the same time, this sodium, so it's called a sodium-potassium pump. So at the same time, you're also moving potassium. And potassium is really big, has a real high concentration inside the cell and a low concentration outside the cell. So I draw a little k for low concentration of potassium and draw the back brackets for concentration. And I could do that for sodium as well. So that means sodium concentration in general is really high, has a high concentration outside the cell in the interstitial fluid, and then it's got a real low concentration here inside the cell, or the intracellular fluid. And so this sodium-potassium pump at the same time will pump potassium out, or it will pump sodium out and potassium in. And it needs ATP to do that. And that's what it shows in the cartoon. So active transport in general requires protein carriers. That's this purple thing. This is a protein carrier. So that means that could be, okay, that could be a little confusing to call it a protein carrier. When it's called a protein carrier, that means the carrier itself, this purple stuff, is made of protein. But what it's carrying are these ions, sodium and potassium ions. So the carrier is a protein carrier, transports substances, in this case ions, that may not be lipid soluble. So lipid soluble, if you were a lipid soluble molecule, that means you could pass freely across the phospholipid bilayer because you would be soluble in this giant hydrophobic layer in the middle. So if you are a molecule and you're not lipid soluble, you can't pass freely across the phospholipid bilayer, you need a protein carrier. A substance may need to move against its concentration gradient, like we've been talking about. You could say against, or you could say up a concentration gradient. If you need to do that, if you need to move against a concentration gradient, you need ATP. Okay. Yes, that's what I just said. ATP is needed as energy to do the transport that we just talked about. So we went over this in the previous cartoon, but let's check out the notes. It's called the sodium-potassium pump. The pump will pump sodium to the ECF, and remember we said we could also call this the interstitial fluid, ECF, or interstitial fluid, and then the pump simultaneously pumps potassium into the intracellular fluid. So let's look at the notes. Binding of cytoplasmic sodium to the pump. That's what it's doing here. Sodium is binding to the pump. And they say the pump protein, so they're just saying the pump is made of protein. That stimulates phosphorylation by ATP. What that means is when these sodiums bind to the pump, this purple pump is protein, and it's gonna twerk just a little bit. It's gonna move around a little bit, and it's gonna open up this site for phosphorylation, to stick a phosphate. Where does that phosphate come from? In this case, it comes from the third phosphate on ATP. That phosphate is gone now, so instead of adenosine triphosphate, now you are adenosine diphosphate. And then Well, it says here, so that when phosphate binds to the pump, causes the pump to change its shape. But really, when the sodium binds, the pump changes a little bit its shape, and then when phosphate binds, it torques its shape even a little bit more. Now this shape change, so the phosphorylation and the shape change causes the sodium, the pump, to kind of do this. So the pump was like this V, and then what it did when it got its 3 sodiums, it went like this. Boop. And now the V flips and opens to the outside, and the sodiums are released. That allows potassium now to come in to its site. And you may notice there's, for the pump, when the pump is working, you get 3 sodiums, Na+, pumped to the outside, and only 2 potassiums pumped to the inside. Okay, so, and then it also shows here some molecular details. The phosphate will leave that site. So when potassium binds, the phosphate leaves the site. When the phosphate leaves the site, then the protein pump goes back to its original conformation or shape. That's the same thing as shape. And then the potassiums are released to the inside, or the ICF. And that now the pump, see how it's open down here? Now 3 more sodiums combined here, here, and here. And the pump can keep working as long as there's ATP present. The pump keeps working. So I can't stress enough how important it is to understand how this pump works, because you're gonna see it for the rest of your scholastic career. Now, maybe when you get into the clinic, you'll never hear the word sodium potassium pump again. I don't know, because I don't work in a clinic. But I know for the rest of your classes, if you don't dig on the sodium potassium pump, you're going to be lost in a lot of classes. So start digging on the sodium potassium pump. All right, more active processes. So there's this thing called vesicular transport, and we've already kind of seen this when we saw the endoplasmic reticulum pinching off a vesicle with a protein in it, and then that vesicle moving to the Golgi apparatus. That was a type of vesicular transport. So a vesicle is just a little transport pod that the cell uses to move things around. So, and they call it a vesicle. I'm not sure the Latin root of that. I should look that up. But anyway, vesicular transport. In this case, we're going to talk about a substance that moves in a vesicle, and it doesn't actually cross the plasma membrane. Okay, so exocytosis is an example of this vesicular transport. So I just said this a second ago. Here's the endoplasmic reticulum. Endoplasmic reticulum buds off one of these little vesicles. It fuses with the Golgi, and then the molecules inside the vesicle get tricked out inside the Golgi apparatus. And then another vesicle pinches off the Golgi apparatus, and that vesicle that's filled with all these proteins that the cell wants to get to the outside will fuse. The Golgi vesicle will fuse with the plasma membrane. Here you can see the fusion. That means just the 2 phospholipid bilayers sort of meld together, they fuse together. And now the material, the proteins that were inside the vesicle, can be exocytosed to the outside of the cell. And it says here moves material. So, and that material are just these little balls, which are, they could be proteins. They could be proteins with sugars stuck on them. That would be a glycoprotein. They could be a protein with a lipid stuck on it. But anyway, that's what materials are. That's exo. So exo is exit. So you're some protein or some material that wants to be, that the cell wants to exit. Opposite of that is endocytosis. That's when you move material into the cell. So check out all these little green balls on the outside of the cell. Yeah, so here's the plasma membrane of the cell. And endocytosis, you can see you form this little vesicle here, and these green balls move into the vesicle, and then the vesicle pinches off from the plasma membrane, and now the vesicle with your little green balls are inside the cell. And in this cartoon, it shows this primary vesicle will fuse with another vesicle, and then that vesicle will exocytose the green balls. But anyway, endocytosis and exocytosis, maybe you've heard of it before, but they're really important vesicular transports and systems in cells. Endocytosis, there's three subtypes we want to talk about: phagocytosis, pinocytosis, and receptor-mediated endocytosis. Really important. Phagocytosis, really important in the immune system. So you'll see that when you study the immune system in 202 and 205 and 218. Pinocytosis, don't talk a lot about that. Receptor-mediated endocytosis is really important in understanding how cells receive a signal on the outside and transfer that signal to the inside of the cell. Okay, so we went over this in previous videos, but it's a review: vesicular transport of exocytosis. So here's the rough ER. So remember why it's called the rough ER, because it has these ribosomes, all these little dots, ribosomes. That's where you make proteins. And remember, the cistern is just this space inside the ER. So you're making these proteins on the ribosomes, and the proteins are made. You get a transport vesicle. It fuses with the Golgi. Those proteins that were made on the rough ER are now in the Golgi, and they're getting tricked out even more. You get a vesicle that pinches off the Golgi, and then the Golgi delivers the proteins that you made to the outside via exocytosis. The second pathway, remember the Golgi vesicle could be here. You could pinch it off, and really you're just making, it doesn't show it, but there could be proteins here sticking out of the Golgi, and those proteins, you don't want to exocytose them. You don't want to put them out into the interstitial fluid. You want to keep them in the plasma membrane. So, and you can see you have fusion of the Golgi vesicle with plasma membrane here, and then this thing will eventually straighten out, and you'll have your fused Golgi membrane with the proteins sticking in the membrane, the vesicle membrane, and now it's part of the plasma membrane. And then pathway 3, the Golgi vesicle could go this way with all its proteins and digestive enzymes, and then those digestive enzymes in the Golgi vesicle would fuse with the lysosome, and now you put all those digestive enzymes into the lysosome. This is cool. This is an electron micrograph of a secretory vesicle. So here's the secretory vesicle from the Golgi apparatus, and so here's the interstitial fluid out here, and then this would be the intracellular fluid here, and the vesicle is releasing its contents right here to the interstitial fluid. More on vesicular transport. So this is the cool cartoon. I worked with a graduate student that studied these things called snares. So here's the ECF, otherwise known as the interstitial fluid. Well, ECF is the extracellular fluid. ISF is the interstitial fluid. Here's the cytoplasm, or the intracellular fluid. Here's a secretory vesicle that wants to fuse with the plasma membrane, and the way it can do that is it gets a signal from this thing called a T snare, and here's the V snare on the vesicle. They recognize each other. So this vesicle basically knows where to go in the plasma membrane. And then you can see here the snare is fused. It's like, yeah, you got docked in the right place. And then the material is exocytosed. This is not on the test, but these snares are kind of cool because they help direct the vesicle to the right place in the plasma membrane. Endocytosis. Endocytosis, extracellular substances, like these green balls, are being engulfed, or you could say they're ingested or they're taken in, or they're endocytosed via this membranous vesicle. So membranous, you should always think if it's got a membrane, it's the phospholipid bilayer or a lipid bilayer. And then once the substance to be ingested, the green balls, is ingested, you're a vesicle, and we talked about this, the vesicle can fuse with a lysosome, or the vesicle could move to the other side of the cell and just release the contents on the other side of the cell. So, and this happens because maybe this is a side of the cell over here, and this is another side over here, and you want to get these green balls from one side of the cell to the other side of the cell, which you do. It does happen. There's a lot of those cases you'll see in 202. And then we said this earlier, there's 3 types of endocytosis. There's phagocytosis, really important. Pinocytosis, don't talk a lot about that. And then receptor-mediated endocytosis. Okay, so this is phagocytosis. That's cell eating. So I think phagos with the PH, I think it's Greek and not Latin for eating, to eat. But the cell engulfs large particles like bacteria or dead body cells. So these could be dead body cells. These green things could be bacteria. And this cell could actually be a phagocytic cell that does phagocytosis for the immune system. And so here's the cytoplasm, or the ICF. Here's the extracellular fluid out here, or the ECF. Could be the interstitial fluid as well. Here's all the things that need to be engulfed. And then look at this guy, bacterium. So this cell, which is probably a phagocyte, will engulf the bacteria, take it inside its cytoplasm or cytosol, and chop it up, and then release the parts of the bacteria that can no longer be dangerous. Pinocytosis is cell drinking as opposed to cell eating. And so it's kind of the same cartoon, but I just made this word up, by the way, a gulpopod. So in cell drinking, you gulp whatever is outside here. It's usually when you're drinking something, you don't usually drink a bacteria. You could drink smaller molecules. So it says the cell gulps droplets of extracellular fluid, and that those droplets of extracellular fluid often contain things that are dissolved in the extracellular fluid, which could be something like a protein or a fat. This is a routine activity for most cells, like cells in the small intestine. They form this, they do this pinocytosis all the time. And then receptor-mediated endocytosis. This is really cool because well, first of all, here's our cell. So this is a cell membrane. This is the cytoplasm of the cell. Here's the extracellular fluid, or ECF, or it could be the interstitial fluid. And here's all these molecules on the outside. And check it out here, though this Y, this is a receptor. Now, this is very specific. The receptor only binds things that fit the shape. And these purple triangles fit the shape of the receptor. So receptor-mediated endocytosis is a method to take up specific molecules, specific ones. These receptor proteins on the membrane surface, like this guy, can only bind specifically, in this case, to the purple triangle, like I said. This is a highly selective process because you're only taking in the molecules you want to take in to your cell. So you only want to take in molecules that fit the Y. This cartoon is a little bit misleading because it looks like you're taking in these green balls too, but you're actually not. You're only going to take the things that fit the receptor inside the cell. And then this just gives you a cartoon of the whole process of endocytosis that we've been talking about.